Hi, my name is Jens-Uwe Ulrich. I'm a PhD candidate at the Hasso-Plattner Institute in Potsdam, Germany, and in this short video I present you our new tool for taxonomic classification of long weeds, which we call Taxor. Microbes are everywhere. They can live even under the most extreme conditions, and we know that the microbiome in and on our body plays an important role in human physiology and diseases. To study this microbiome, metagenomic sequencing has become a key technology. A typical workflow for metagenomics sequencing looks as follows. We take, for example, a sample from the human gut, and then we extract all the DNA in that sample and sequence it on a sequencer. As a result, we get a large set of sequencing reads, and then we want to know for each of these reads from which reference genome or from which species they originate. And then finally we can do something that is called taxonomic profiling, where we simply calculate the abundance of the species. The biggest problem of taxonomic classification is the exponential growth of reference genome databases which we need for the classification of the weeds. And due to this, the current methods have large computational requirements. Thus we need new data structures for fast and memory-efficient querying of long weeds. This searchable database index should be able to contain thousands of references, but also fit into the memory of a laptop. And we need a small index that also guarantees a low false positive rate. At the end, it would be nice if we could enable real-time metagenomic profiling of long weeds even on small laptops. In order to achieve these goals, we developed a new data structure that we call hierarchical interleaved XOR filter. Based on the previous work on XOR filters, we first developed an interleaved XOR filter. Then we extended this data structure to a hierarchical interleaved XOR filter in order to avoid using more space than necessary when the references in the database are highly divergent in size, as for example using viruses and also fungi in the same database. This new data structure is implemented in our new tool, which we call Taxor. Taxor offers the following features. First, we have a significant space saving over Bloom filter-based approaches. We offer fast taxonomic classification of long weeds by using also open canonical zinc-mares as a k-mere selection scheme. For the final profiling, we use a three-step filtering of the initial search results and refine the classification results with a standard EM algorithm. In order to evaluate our new tool, we compared it with 5 state-of-the-art taxonomic profiling tools. And to ensure a fair comparison between the tools, we used exactly the same reference database for all the tools. So at the end, we used a custom database that consists of 21,003 reference genomes, and we used a comparable k-mere selection scheme and parameters for all the tools. Then we took 3 real mock community dataset of long weeds, and we measured the weed classification metrics on different taxonomic levels. Finally, we did a benchmarking of the computational requirements for building the database index and querying of the weeds. Our benchmarking with regards to the weed classification metrics showed that Taxor has the highest precision for the 2 nanopore datasets Zymo R10-3 and Zymo Q20. Only for the bio dataset KMCP has a higher precision than Taxor. However, KMCP has the lowest recall of all tools for all 3 datasets. On recall level, only MetaMaps, which is the only other long weed taxonomic classification tool here, has a higher recall, closely followed by Gannon and Taxor. When we look at the F1 score, we see comparable results for Taxor and MetaMaps, and for the F0.5 score that prioritizes precision over recall, we see that Taxor slightly outperforms MetaMaps and is far better than the other tools on the species level. But our main intention was to reduce the computational requirements for metagenomic classification. So we did a benchmarking, first building the database and then querying the first Oxford Nanopore datasets against this reference database. And what we see is that Gannon is the fastest tool for building the custom database index, but it takes a lot of memory. Here 124 gigabytes. We see that Taxor is as fast as Kraken 2 when building the database, but needs just half of the memory as Kraken 2. And Taxor has by far the lowest index size of all the tools. And this is the main advantage when we query the read sets against the database index. Here we see that Taxor is as fast as Gannon and Kraken 2, and also has by far the lowest memory footprint of all the tools. Finally, we can conclude that Taxor improves long-read taxonomic classification, and that our hierarchical interleaved XOR filters enable fast queries by also reducing the memory requirements by more than 50%. These results show that Taxor can enable real-time long-read taxonomic profiling on limited computing resources. Taxor's source code and precompiled binaries are already available at the Git repository from the Winar Lab at the Data Analytics and Computational Statistics Chair of the Hasso-Plattner Institute. And if you'd like to hear more about hierarchical interleaved XOR filters and Taxor in general, I'm happy to see you all at my HitSec talk on the 26th of July at the Lumiere Auditorium.